Okay, guys, so in the last slide, we talked about the lytic cycle in which the virus goes into a cell, it infects it, and it replicates, the cell replicates so much virus that it ends up killing the cell. This is the other pathway called the lysogenic cycle. So in this type of viral reproduction, the host cell makes copies of the viral genetic material indefinitely without necessarily killing the cell. The virus incorporates its DNA into the host cell's DNA or chromosome. So it becomes a permanent part of the host cell DNA. So every time that cell divides, that viral DNA is going to be replicated along with the host cell's own DNA. So you can see this is a way a virus replicates just by the cell replicating itself. At least its genome or its DNA is being replicated. It doesn't necessarily make viral proteins though right away. And so it doesn't kill the cell right away. So that is a way for a lysogenic virus to remain dormant or inactive for some time. But remember, every time that cell is reproducing, that viral DNA is also being reproduced as well. So here we compare the lytic cycle on the left versus the lysogenic cycle on the right. So if you go to the upper left-hand corner, you see a bacteriophage attaching to the cell, and it injects its DNA, and we have the bacterial chromosome there. And then we go down to the middle, and certain features, now we've got the viral DNA in blue and the host DNA in brown. Remember, the capsid stays on the outside. So certain features determine whether the lytic cycle goes or the lysogenic cycle goes. So we'll go on the left side and look at the lytic cycle. In the lytic cycle, viral proteins are being made and viral progeny are being produced. So we've got lots of copies of the virus inside the cell. And then it goes up and the host cell bursts, releasing new virus. And remember, all those viruses now can infect new cells. However, if we go back to the very middle where it says certain features determine, let's go to the right and say the lysogenic cycle is entered. Well, then the viral DNA gets incorporated into the host cell's chromosome. And we call that a prophage. Pro means before. So it's before a phage. So it's just the DNA incorporated into the chromosome. The viral DNA is incorporated, forming a prophage. And then every time that bacterial cell divides, by binary fission it's called, the viral genome is also copied and passed on to daughter cells. And then many cell divisions may occur, producing a large population of bacteria that are infected with the prophage. It's a pretty sneaky way, actually. And then we have a daughter cell with the prophage, and it can go through the whole cycle again. So in the lysogenic cycle, the prophage is the viral DNA embedded into the host cell's DNA. The prophage may remain part of the host cell for many generations before becoming active. So eventually, certain environmental conditions like chemicals or radiation may trigger the switchover from the lysogenic, in which it's dormant, to the lytic cycle, in which it's virulent. And remember, virulent means it causes great harm to the cell. And if Screencastify doesn't cut me off, let me try to get through this slide. Retroviruses. So in humans have this too. We just talked about phages, which apply to bacteria. But retroviruses like HIV, they have RNA as their genetic information rather than DNA. And these viruses have an enzyme called reverse transcriptase. So we go, instead of from DNA to RNA to protein, we go from RNA to DNA, back to RNA, then to protein. So this transcribes RNA template into DNA. So we're going from RNA to DNA. That's reverse. The newly made DNA then enters the cell's nucleus and integrates it into the DNA of a chromosome. And in this way, just like a prophage, this is called a provirus. The retrovirus may remain dormant for some time. It will eventually become active, causing the cell to make new viruses and causing the death of the host cell. Retroviruses are responsible for some types of cancer as well. And like I said, the HIV